Thank you for that introduction. You may have heard in the media, in the news, in the newspapers about this crazy idea known as intelligent design. What I want to do through the course of this presentation is to try to clarify for you what intelligent design actually is, what it says, and more importantly, what it doesn't say, and to dispel, hopefully, some popular misconceptions that abound concerning this theory of intelligent design. So let me list just a short outline of this presentation. I'm going to introduce intelligent design as a concept. We're going to talk about DNA and the fact that it contains information. We'll talk about molecular machines in the cell and how they evidence intelligent design. Talk about some of the common objections to design in biology and how we might address those objections. And finally, we'll draw some conclusions. So let me start by introducing intelligent design. How many of you have heard of intelligent design? Show of hands. Most of you? That's great. Would anyone like to try to give me a definition of intelligent design? Any volunteers?
It's in the name.
Say again?
It's in the name. Design that is intelligent by nature and by faculty.
Uh-huh. So what would you understand by the scientific theory of intelligent design? Anyone give me a definition of how? I mean, let me give you a popular definition that's found in we're often given by the media and pop culture. Intelligent design is the idea that living systems are too complex and improbable to be explained by the process of evolution, and therefore require invocation of a supernatural being to explain their existence, such as God. How many of you would buy into that sort of definition of intelligent design? Show of hands. Yeah, quite a few of you. Okay, anyone see any problems with this definition? Anyone disagree with that? No one? Okay, one problem with this definition is, of course, that it presents and sets up the theory of intelligent design as an argument based on ignorance. It's a God of the gaps. It presents it just presents a negative critique of naturalistic explanations. It doesn't actually present anything positive as an alternative to design. It just says, well, we can't explain it as a result of evolution and blind natural processes, and therefore we'll just plug in God or an intelligent designer to explain the origin. And so as you come up with new naturalistic explanations, intelligent design as an idea has to retreat with each new advance in scientific discovery. And of course, that's a problem. Let me propose a better definition of intelligent design, which I think is a better way of framing it as more of a positive inferential case for design in the world. Intelligent design is the idea that we can infer from patterns in nature that some features of living systems are best explained by an intelligent cause rather than a stochastic process involving chance and necessity. So you can see immediately that I've not presented my argument merely as a critique of evolutionary processes, but I've actually presented a positive argument for design. Now you might ask, well, how can I say that intelligent design is the best explanation for features in biology? We'll come on to that through the course of this presentation. Let's talk about the alternative that is the mainstream view in modern biology. This is Darwin's theory of evolution by natural selection. Here you can see, of course, Darwin's tree of life. And this, of course, represented for Darwin the idea that all forms of life that are alive today on planet Earth are descended with modification from previous ancestors, previous organisms. And ultimately, if you trace all organisms back through about more than 3 billion years, you come to a single progenitor of life, which was the last universal common ancestor of all life that we see on Earth today. Now, the theory of evolution consists of 6 main elements or components. It can refer to the change in allele frequencies in a population over time. Now, an allele, as you'll know if you're a biologist, is a gene variant. And the idea is that as the frequencies of alleles change in a population over time, natural selection preserves the successful gene variant combinations, and the successful combinations of gene variants then are passed on to one's progeny and become fixed in the population and so on. So the changes in alpha frequency in a population over time, that's not controversial. Everyone acknowledges that. Then there is the concept of speciation, the idea that by various mechanisms, there are perhaps geographical barriers that are set up that separate and isolate a particular population. It can lead to new species. And another concept that comes under the banner of evolution is the idea of common ancestry, which I've already alluded to, the idea that you can trace organisms back to common progenitors, common ancestors. And of course, speciation is not controversial. Common ancestry, intelligent design doesn't necessarily challenge common ancestry. We'll talk about that. Gradualism is another idea that is associated with evolution, the idea that life evolves and changes very gradually by numerous slight successive variation. Life doesn't take sudden leaps and bounds. Natural selection, the idea that we have this force which doesn't create anything, doesn't generate complexity, doesn't produce complexity, but nonetheless preserves the complexity, or the variations rather, that are produced by random genetic mutations. Now, genetic mutations occur by various mechanisms. Basically it refers to changes that occur in DNA, and perhaps as a result of copying errors as the DNA is copied, and the DNA polymerase as it's going along might make a copying error, and that might be preserved by natural selection or even by genetic drift, which basically is a more random process. And that basically natural selection provides the grist for the mill of natural selection. So most of these concepts actually are not necessarily incompatible with intelligent design as an idea. The change in alpha frequency in population over time, that's not necessarily incompatible at all with intelligent design. Speciation, not incompatible with intelligent design. Common ancestry often is not actually incompatible with intelligent design. You could have evolution which is guided by an intelligent agent. And intelligent design proponents actually disagree about the extent to which common ancestry is true. For example, someone like Michael Behe at University of Lehigh, who's a biochemist who supports intelligent design, agrees with the notion of common ancestry, even though he disagrees with the broader, the neo-Darwinian theory of how evolution occurred. Gradualism isn't necessarily inconsistent with intelligent design, but I think that gradualism is more expected under the Darwinian model. Natural selection, again, natural selection occurs. We see antibiotic resistance, we see insecticide resistance, we see natural selection preserving successful phenotypes or the expression of genetic variation. And then the most controversial part for an intelligent design proponent like myself is this last one, random genetic mutations, indeed the sufficiency or the adequacy of random genetic mutations in generating the complexity necessary for the origins and evolution of life as we know it on Earth. And so intelligent design proponents might disagree about common ancestry, but we're unified in critiquing the causal efficacy of this random genetic mutation mechanism. So how do we detect design? Here we see a cliff face. And you look at this cliff face, and how many of you would say this cliff face was designed by an intelligent designer? Any of you know? Well, there's nothing to see here. There's no design to see here. You see immense improbability, right? This is an immensely improbable conformation. So from this we can see that improbability doesn't give us an inference to design, right? You can have immense improbability without inferring design. But how about this one? Right? How many of you would recognize the product of design here, right? This is Mount Rushmore in South Dakota. And you can immediately see that this is the product of design. You see the president's faces carved into the rock. So can anyone give me a reason why they infer this is the product of design, whereas this is the product of wind and erosion? Why would you be able to empirically say that this is the product of design? Anyone have any suggestions or ideas? Uh-huh. Mm-hmm. Good. So let's press on. You may have seen the movie Contact. This is Jodie Foster here. The movie Contact was based on a novel by Carl Sagan, who was a famous popular scientist and cosmologist. And basically the idea in the movie is that it's based around the SETI program, or the Search for Extraterrestrial Intelligence. And as the SETI researchers were doing their work, they discovered radio signals from outer space, and basically it was specified the first 20 prime numbers. And so they inferred that this is not the product of stochastic noise, but is in fact the product of design. Some extraterrestrial civilization is trying to communicate. But why would this allow us? Why would this allow us to detect the product of design? Because it's no more improbable than just any other random sequence of numbers. Which of the following is designed, right? The first is, they're both 30 characters, they're both equally improbable, they're both equally complex. But of course you can see the latter one there's something different about it. It actually conforms to some independently given pattern. In the beginning was the Word, of course, and we're all familiar with from John 1, verse 1. Now, I want to introduce to you the concept of specified complexity. So this is basically the criteria which we employ in order to infer the products of design in the world. Specified complexity describes phenomena where 1, the probability of a phenomenon being produced by chance alone exhausts the available probabilistic resources, and 2, the phenomena conforms to some independently given pattern, right? You'll notice that in all the examples I gave, you have those two criteria met. You have complexity and probability, but you don't only have that, you also have conformity to some independently given pattern. Let me give you an illustration. This is an ATM machine, and I'm sure you've all used ATM machines. You plug in your bank card, and you dial your 4-digit PIN number, and the bank will typically give you 3 opportunities to get the PIN number correct before it locks you out of your account. And there's what's called a combinatorial space, or the number of ways of dialing in a 4-digit number, of 10,000 possible ways of dialing in your 4-digit number. Now, out of all those possibilities, how many gives you the PIN number you need for your bank card? 1 number out of all these, 1 4-digit number. So you have a 1 in 10,000 chance of getting the number right just by chance if you only have 1 attempt. If you have 3 attempts before the bank locks you out of your account, then is it more likely than not, or less likely than not, that by chance alone you'll be able to find the right PIN number, right? Surely it's immensely more likely that you will fail and not succeed in guessing and randomly acquiring the correct PIN number. So the PIN number there is specified complexity. Given the number of trials that you have, the probability of attaining the correct PIN number is astronomically small, and it also conforms to some independently given pattern. So those two criteria coming together give us the inference to design. Now, how do we reason about the past? Does anyone recognize this man? Anyone who this is? This is Charles Lyell, famed 19th century geologist. Wrote a very famous book in the history of science, a landmark book called Principles of Geology, being the attempt to understand changes in the Earth's surface with reference to causes now in operation. That was the subtitle. And Charles Lyell was a great influence and inspiration, in fact, to Charles Darwin himself when he was writing The Origin of Species. Charles Lyell insisted that if you want to explain an event in the remote past, then one should let one's present experience of cause and effect guide one's search for the best explanation. And intelligent design became known as the historical abductive method of scientific reasoning. And intelligent design basically employs this historical abductive method, the same method that was employed even by Charles Darwin in the publication of the Origin of Species and the formulation of his theory of evolution by natural selection, to the intelligent design hypothesis. So we find certain patterns in nature, certain patterns in biology, or in cosmology or physics, however it happens to be, and we know in every realm of experience that this particular type of pattern or phenomenon is uniformly associated with intelligent causality. And so when we find this particular pattern and particular information content in the cell, we infer that the most likely or best or most causally adequate explanation for that information content is that it arose by virtue of an intelligent agent, because information is uniformly and habitually associated with conscious activity, whether you're looking at a blueprint or a hieroglyphic inscription or the paragraphs of the book or a page of text on a computer screen, whatever it happens to be. So let me advance this argument further and look at some of the information-bearing properties of life's hereditary molecules of DNA and RNA. DNA contains information. Scientists have discovered, and have known for a long time, that along the spine of the DNA molecule are characters which function like alphabetic letters. They're represented by the letters A, C, T, and G, and these basically represent chemical subunits which which specify different jobs in the cell. There's about some of our DNA, about 1.5% of it, specifies how proteins fold into their respective structures in order to perform jobs in the cell, whether that be taking part in some macromolecular machine or taking part in some enzymatic pathway or whatever it happens to be. And there are different codons in the cell. There are three-letter words that are called codons, and each codon specifies a different amino acid. And there are 64 different codons, 20 amino acids, and you have this codon to amino acid mapping system. And so you have this genetic code, a language, if you will. Languages come from intelligent causes, not from stochastic, blind material processes. So as I said, the information DNA directs the construction of proteins. This is what's called a ribosome, this structure here. And you can see these amino acids. This is methionine, arginine, serine, being brought in by transfer RNAs. These are called transfer RNAs here. And along here, we can see a transcript of messenger RNA. Messenger RNA is the intermediary between DNA and proteins. DNA is taken and it's transcribed into a single-stranded molecule called messenger RNA, and it's taken to a ribosome, which is a two-part chemical factory. And these amino acids are sequenced in accord with the instructions on this transcript, and the sequential arrangement of these amino acid subunits will then determine how the protein collapses into a three-dimensional structure, which then specifies its particular job in the cell. Can you maybe click this video? This is a brief video by my friend Stephen Meyer, which animates this process.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
So the idea is that you see this complexity, this design, this exquisite case of engineering prowess at its finest in the cell, and you have this DNA language system, this DNA coding system. You have this informational retrieval system, which I think is best explained by design. This is the genetic code. This is the codon to amino acid mapping system. So this represents the language that we find, the language of life that we find in biology. Now, as already been alluded to, the sequential arrangement of amino acid subunits determines how a protein will fold into its respective three-dimensional conformation. So down here at the bottom, you see the amino acid primary structure. You see the different amino acids here in their primary structure, the linear form. And then you have the secondary structure here, like alpha helices and beta strands, which form beta sheets. And then you have the tertiary structure, which is where you have alpha helices and beta sheets coming together. And then you have the quaternary structure, where these form domains of proteins, and then these domains pack together to form a quaternary structure. And this gives the protein its different functions within the cell, taking part in biochemical pathways or contributing to molecular machines and so on. Many molecular machines have dozens and dozens of these protein components that are responsible for making their function possible. Now, how do we explain the origins of protein folds, right? It's one thing to explain the macromolecular machines, which require multiple proteins to work together in a very coordinated fashion, which we'll come on to in a little bit later. But how do you actually explain the individual protein components that are necessary to contribute to these machines or biochemical pathways? Well, the prevalence of functional folds turns out to the prevalence of functional folds in combinatorial sequence space turns out to be very important in addressing the question of the adequacy of the mutation selection mechanism to explaining and accounting for this phenomenon. As I mentioned, there are 20 or so different amino acids that are used in cellular proteins, and this means that for a modestly length protein of, say, 150 amino acids, that's quite modestly length. There's many proteins that are much longer than that. There are 20 to the 150th power of possible combinations. That's 20 times 20 times 20 times 20 times 20, all the way up to 150, right? So the question we want to ask is, out of all these possibilities, how prevalent are functional protein folds in combinatorial space, right? The English language conveys meaning precisely because the ratio of functional to non-functional sequences of characters in the English language is astronomically small. And so we recognize when you read lines of English that it came from an intelligent designer, not from random typing errors on a keyboard. And so the question is, does biology, do proteins actually reflect this same phenomenon? Because if it did, if the ratio of functional to non-functional sequences within this vast array of combinatorial possibilities was astronomically small, then it would be very difficult to conceive how you could go from one island of function to another by the mutation selection mechanism, given the time allowed since the origin of life. Or even to get life started, because life itself requires a minimum 200 or 300 proteins or more. So here is a case where intelligent design and Darwinism both make competing predictions. Under the Darwinian paradigm, Darwinism would predict that you would have a relative abundance of stable and functional protein folds within combinatorial space, whereas intelligent design paradigm would predict that you'd find an astronomical rarity of functional protein folds within this vast array of combinatorial possibilities. So what turns out to be the case? Well, here's one example. This is a paper published in the Journal of Molecular Biology in 2004 by a friend of mine called Doug Axe, who currently works at the Biologic Institute in Fremont, Washington, but he at the time worked at the University of Cambridge. He was doing his postdoc, and this is published 2004, and basically he conducted what's called a site-directed mutagenesis experiment, where he randomly altered the protein's amino acid composition in order to attempt to elucidate the ratio of functional protein folds within combinatorial space. And so this is one case where this has been empirically tested. It's often claimed that intelligent design doesn't do peer-reviewed research, so that's completely bogus. Here's one example, and there's many, many more as well, just to give this example. Here's his conclusion. Combined with the estimated prevalence of plausible hydropathic patterns for any folds and of relevant folds for particular functions, this implies the overall prevalence of sequences performing a specific function by any domain-sized fold may be as low as 1 in 10 to the 77th power, adding to the body of evidence that functional folds require highly extraordinary sequences. So to put that in context, there's only 10 to the 65 atoms in the galaxy. There's about 10 to the 80 elementary particles in the observable universe. So this is an astronomically small prevalence of functional folds within combinatorial space. And this isn't the only estimate. There are other estimates. Robert Sauer, for example, back in the 90s at MIT did some great work. Doug Axe actually built on that. And Doug Axe and his colleague in Washington, Ann Gager, have done subsequent work which has confirmed this even further. And there were tests by other scientists which have conformed to this data as well. So this presents a probabilistic resource problem. So there are approximately 10 to the 80 elementary particles in the observable universe. Since the Big Bang, roughly 10 to the 16 seconds have elapsed. And the time it takes light to traverse the smallest distance, i.e. the Planck length of 10 to the minus 33 centimeters, is 10 to the minus 43 seconds. And so by multiplying those factors together, it can be estimated that the maximum number of events that could have taken place in the observable universe since its origin is 10 to the 139th power. Now, I think that plausibly you can rule out the chance hypothesis at levels far lower than that, but that can give us a universal probability bound. And this presents a needle in a haystack problem. Locating the functional sequences within the vast set of combinatorial possibilities by a random search is like finding a tiny needle in an enormous haystack and having a very limited time to search. Now, the problem extends deeper. When molecular machines require multiple cooperating proteins in order to perform their functions, then these probabilities multiply exponentially, right? This is, here I've shown a sort of diagrammatic illustration, very simplified drawing of DNA replication, how DNA is synthesized within the cell and copied by DNA polymerase. It requires other enzymes like helicase and topoisomerases and ligases and so on. And you see this concert of macromolecular machines which each contribute to, in very specific ways, in very coordinated ways, to produce this function which is necessary for cell division, necessary for DNA to replicate itself. It requires many, many, many proteins. And so the probabilities just multiply exponentially as you build these macromolecular machines and as you construct biochemical pathways in the cell. This is, I'm just going to show a short video clip this is from an Illustra Media documentary. It's a pro-intelligent design documentary produced in America, and basically the documentary was about an event in the history of life known as the Cambrian explosion. The Cambrian explosion is an event which occurred about 530 million years ago, which documents the abrupt, explosive emergence of many of the animal phyla. Animal phyla include things like arthropods and echinoderms and so on. And you see an explosive diversification within the history of life, which requires new information content. And this particular clip looks at one metaphor that has been put forward for the neo-Darwinian paradigm that's been put forward by Richard Dawkins in his book Climbing Mount Improbable, where basically he said, from the front side, the mountain might look like a sheer cliff, But the Darwinian story is that if you go back, if you look at the backside of Mount Improbable, you have a slowly sloping hill leading all the way to the summit. And so you can scale this complexity not in one giant leap, but by numerous successive slight variations. And so Richard Dawkins put forward this metaphor of climbing Mount Improbable. And this documentary is just going to look at that and see how tractable it is. Click on that. Thanks.
Richard Dawkins, the famous Oxford evolutionary biologist, has illustrated how the Darwinian mechanism works using a metaphor he calls climbing Mount Improbable.
From the front side, the mountain is a sheer cliff that could never be scaled in one giant leap. For Dawkins, this represents the impossibility of creating a complex animal by chance alone. Yet Dawkins also envisioned an alternative route up the backside of Mount Improbable, a long, gradually sloping trail of small steps leading all the way to the summit.
According to Dawkins, that's how you'd climb the mountain, and that's also how you'd build a Cambrian animal, one small step at a time. What chance alone can't accomplish in one blind leap, natural selection can accomplish through the cumulative effect of many small incremental steps.
In theory, each step corresponds to a small unit of biological change, a new gene and its protein product. But do mutations and natural selection have a reasonable chance of producing even one protein in the time available? Since 1992, molecular biologist Doug Axe has examined this question.
There's a story that's being told, and there's a appeal in the case of Darwinism to random mutation and natural selection as being, in vague terms, the mechanism. But if you look at the detail, what kind of mutation can accomplish these transitions? And there, it's important to realize that the one area where we can really nail this down is at the single protein level, where you can actually measure it. And if you look at protein structures, to get a substantially new protein fold is prohibitively difficult.
Each of the thousands of different proteins in nature is actually a chain made from a specific combination of 20 different amino acids. The sequential order of these chemical building blocks is crucial, for if they are arranged correctly, the chain folds into a functioning three-dimensional molecule. But if the amino acids are incorrectly assembled, no protein will form. If proteins are indeed rare among the possible sequences of amino acids, what are the odds that mutations would stumble upon a functional combination of chemicals from the vast number of alternatives? To find out, Axe randomly altered the structure of an enzyme protein comprised of 150 amino acids.
You've got the protein 150 amino acids long, then you've got 20 to the 150th power possible ways of arranging the amino acids. Out of all those possibilities, how many are functional and how many are gibberish?
If you do the experiments and you analyze how much information is required to get, say, a new protein fold, it's just far beyond what you can get by random mutation and natural selection.
How far beyond? Axe published his findings in the Journal of Molecular Biology. He determined that among all the possible amino acid combinations, the probability of generating just one short protein by mutation is roughly 1 in 10 to the 74th power, or one chance in 100 trillion, 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 trillion.
To put that in context, there's only 10 to the 65 atoms in the entire galaxy. So to build a new functional protein by selection and mutation within the time allowed for the Cambrian explosion, what you're essentially having to do is equivalent to a blindfolded man looking throughout the entire galaxy for one marked atom. So what we're talking about is searching for a tiny, tiny needle in an enormous haystack and having a very limited time to search.
So, on the question of something like the Cambrian explosion, there does not appear to be any way that unguided random mutations can accomplish what needs to be accomplished to explain new functional proteins. And certainly by extension, wherever in the history of life you would need to have multiple new protein folds, the probabilities multiply. So there's no reason to think that this is plausible.
And I highly recommend this documentary if you want to learn more about the Cambrian explosion and how it typifies the severe problem that neo-Darwinian evolution has in explaining the origins of biological complexity. But you might say, well, there's still a chance, right? You've got this immense improbability, but it's still possible. You may have seen this clip from Dumb and Dumber. Do you want to just click on that for me? Thank you.
What are my chances?
Not good.
You mean not good like 1 out of 100?
I'd say more like 1 out of 1,000,000.
So you're telling me there's a chance?
So I call this rejoinder the Dumb and Dumber response. Well, there's still a chance, right? One way around this, some Darwinian biologists propose, is called the method of exon shuffling, which is basically a hypothesis that fundamentally new protein folds can be created by recombining already existing domains, which is thought to be accomplished by basically moving exons, that's protein coding regions of DNA, from one part of the genome to another by a process called exon shuffling. There's different ways that this can be mediated. One is by elements of mobile DNA, or so-called jumping genes. Another way is by crossover during meiosis and recombination. And there's a number of problems with that. I can talk about it later in the Q&A if you want to. But I don't think it works. I think that you have to explain interface compatibility, and there's been some experimental research which I think shows its implausibility. I'm happy to go there later if you want to. Talk about molecular machines in the cell. Molecular machines, of course, are machines, nanomachines, which demonstrate and display and exhibit engineering prowess at its finest. Just to give you a sort of brief tour of the cell to show you the sorts of machines that we're looking at in biology, here's a short animation.
I'm finally just beginning to grasp the complexity of the cell. Are there systems within the cell that go well beyond Darwinian evolution? Some type of cellular technology that drives adaptation, replication, quality control, and repair? What if these new mechanisms have massive design implications? Well, I say, so be it.
And that animation is taken from Expelled, which is a movie produced about some of the persecution that intelligent design scientists and researchers and sympathizers, not just in the sciences but in other disciplines, have encountered, but also present some of the case for intelligent design. That's a beautiful animation that was produced for the purpose of that particular documentary. You may have heard of the concept of irreducible complexity. How many of you are familiar with the concept of irreducible complexity? Some of you. Okay, good. So Charles Darwin, in The Origin of Species, put forward a prediction or test that his theory made. And he said, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Now, of course, I would quibble with the use of the word could not possibly, because anything's possible, right? But not everything's plausible. right? You're telling me there's a chance. Well, it's possible, but not plausible. But this is the prediction that Charles Darwin put forward, that if you could show that a particular complex organ existed that couldn't be put together by numerous slight successive modifications, variations, his theory would absolutely break down. And then in the 1990s, at Lehigh University, this man came along, Michael Behe, and he published a very famous book called Darwin's Black Box. And he put forward this idea, or popularized this idea, that he called irreducible complexity. And he said, by irreducible complexity, or by irreducible complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. And his contention in this book, and the contention of other ID scientists was and is that there are many systems like this in biology, dozens and dozens, hundreds of them. His most famous example, his favorite example, and probably my favorite example, is the bacterial flagellum, which is basically a rotary motor that propels bacteria through liquid. In some species, like in Vibrio cholera, for example, these rotary motors spin at speeds of up to 100,000 RPM with near 100% energy conversion efficiency. They're far beyond human technology and human engineering. They can change. It only takes a quarter turn for these systems to change gears and go the opposite direction. They're built in with a signal transduction circuit. They actually build themselves, so they're actually unlike man-made technology, which is put together by men. They actually build themselves based on instructions. They build themselves from the inside out. And you can see many components which resemble man-made motors. You can see a universal joint or a hook here. You can see this is the propeller, or it's called the filament. You can see the motor has a stator, has a rotor that rotates within the stationary component of the stator. And it's embedded within the cell. This is the periplasmic space. This is gram-negative bacteria. Gram-negative bacteria have two membranes, an inner membrane and an outer membrane. And this particular one, there's so many different flagella in different species of bacteria, and they all have different. They're all arranged in slightly different ways. And the sort of typographic, the model system or the paradigm system is found in species like Salmonella enterica or Escherichia coli, the gamma proteobacteria, as they're called. That, by the way, is actually a photograph or an electron micrograph of one of them. So that's just to show you that's actually an actual bacterial flagellum. It really resembles a man-made motor in many respects. This is just an animation showing the bacterium swimming along. They rotate in bundles typically, and this is the signal transduction circuit here. As you can see, chemical signals coming in, and basically it causes it to switch direction. And what happens is, in response to chemical stimuli or chemical repellents, whether it be attracted towards glucose or repelled by toxins, it causes the flagella to switch direction. They stop going anti-clockwise and start going clockwise, and causes the flagellar bundle to break apart, causes it to tumble in place and reorient itself in response to chemical stimuli or attractants. It's an elegant and beautiful system. This is just an animation of how these bacterial flagella build themselves. Here we go. So here it's going to punch a hole through the membrane using a protein called FlagJ. These are capping proteins which are dispensed with. It's the hook region being built. It's the filament capping protein called FlyD, which is absolutely essential. You knock it out, you lose these flagellum subunits into the medium. So these things actually build themselves. I mentioned rod penetration, so the rod structure actually has to penetrate a hole through the outer membrane to allow the filament to grow. So you have 4 rod proteins, and you have the rod cap protein, which is called flag J, which is bifunctional. It has a C-terminal domain, which possesses hydrolyzing activity, and it has an N-terminal domain, which possesses the binding affinity for rod proteins. So the hydrolyzing domain basically is what punctures a hole through the outer membrane to allow progression of the assembly process. It's essential in gram-negative bacteria. Gram-positive bacteria only have a single membrane, so they don't require that. And besides the 4 genes that encode rod subunit proteins, more than 10 genes are necessary for rod assembly. You have hook length control. Hook length is consistent to 55 nanometers across all bacteria, and the hook length is determined by an elaborate molecular mechanism. There's a number of different models for how hook length works. One of them is, so the hook acts as a universal joint. There's 2 proteins involved, fly K and flu B, this is known. And one popular model for how hook length works is fly K is secreted intermittently and acts as a molecular ruler that actually literally measures hook length. It's an incredible piece of technology. The N-terminal domain of fly K goes, this is how it's supposed to work, goes through the channel and interacts with flag D, while the C-terminal domain forms a globular structure and stays in the cytoplasm. When the N-terminal domain reaches a maximum length, it transmits a signal to the C-terminal domain to interact with flu B, the secretion gate, and this causes a conformational change which occurs to switch substrate specificity. So that's how you get the particular hook length, and also that's linked to the timing as well of filament assembly, as I'll talk about in a second. Filament assembly. So this is the protein structure I talked about, the filament capping protein called FlyD, which has been described as the most dynamic of all protein structures. It has 5 leg-like extensions. It has a pentagonal-shaped disc and 5 leg-like extensions which extend from it. And basically, as you can see in the illustration, it rotates. And basically, as the fly C monomers, or the filament monomers that form the propeller, have to shoot through a 2 nanometer tube, basically a channel that's 2 nanometers in diameter. And they have to be unfolded for insertion into the channel, and they subsequently have to be refolded at the distal growing end. And so this chaperone molecule, this protein, is responsible for ensuring that it folds correctly, basically rotates, and because of this asymmetry, there's always a crevice that allows the protein to fold into the right structure. It's an amazing molecule. In fact, there's no known homologs of this system outside the flagellum, and also flag J, the rod protein, the rod capping protein, has no known homologs outside the flagellum, and there are other proteins in the flagellum that have no known homologs. Even some of these are actually essential to flagellum assembly and function. So now you might ask, how does the flagellum know to assemble itself in this particular order, right? In this particular sequence of events? How do we get the order right? You don't want to start constructing the filament, the propeller, before you've got the hook basal body apparatus in place, right? The base of the propeller. Well, you have this what's called a gene hierarchy, or a transcriptional hierarchy. And I'll try and simplify this. You have basically in the model system, which is like the gamma protein bacteria like Salmonella, Escherichia coli, you have 3 classes of genes. You have the early genes, middle genes, and late genes, or class 1, class 2, and class 3. And the class 1, these are called operons, are basically, and we have a sequence of a series of genes which are regulated together. And these proteins FLU-D and FLU-C come together to form what's called the transcriptional activation complex, which then goes and activates the class 2 genes, the expression of these class 2 genes. And so you have, there's about 35 genes here over 8 different operons, and these basically encode the hook basal body construction. And among them are 2 proteins called flag M, and there's another gene called fly A, which codes for what's called a sigma factor. It's called sigma 28, and basically you have as sigma 28 then activates the expression of these late genes, these class 3 genes, but until it's ready to do so, it's sequestered or inhibited by this protein inhibitor called flag M. At the appropriate moment, when you have the hook, when the hook reaches the correct length, you have this conformational change, which I mentioned, which facilitates the switch in substrate specificity and allows flag M to be exported out this needle-like syringe called the type 3 export apparatus. And it's exported from the cell, and then sigma 28 here is then freed up, it's active, because it's no longer inhibited, and then it goes and activates the expression by binding with the RNA polymerase complex of the class 3 genes, which encode the hook basal body apparatus, as well as what's called the chemotactic proteins, which basically are the signal transduction apparatus of the flagellum. So you have this elegant self-assembly apparatus which builds these things from the inside out. It's an exquisite piece of technology, a piece of engineering prowess. Another molecular machine is DNA replication. DNA replication, of course, involves many molecular machines, protein machines. I'm just going to show a brief animation of this, if you want to click this.
Prior to cell division, the genetic information which is stored in chromosomes must be copied in a process called DNA replication, and the duplicated DNA separated evenly between 2 cells. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in the large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the 2 strands of DNA separate and then serve as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming 2 replication forks. In humans, numerous replication bubbles are formed. Let's zoom in on one of them. Some of the proteins that work together at the replication fork are shown here. Here, DNA helicase unwinds the 2 strands, and DNA polymerases type 3 build new strands of DNA. A critical component of the DNA polymerase III protein is the DNA clamp protein, which prevents the polymerase from dissociating from the template strand. Thus, the process of DNA replication is called semi-conservative. Each new double helix contains 1 original DNA strand and 1 new DNA strand. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously and forms from its 5' end to its 3' end. The orientation of the other new strand, the lagging strand, is opposite to the working orientation of DNA polymerase III, and thus the lagging strand must be built in fragments, called Okazaki fragments, in the 5' to 3' direction using multiple primers. First, let's focus on the leading strand. DNA polymerase III builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Because of the opposite orientation, replication of the lagging strand is more complicated. To initiate synthesis, an enzyme called primase reads the DNA strand and produces a short segment of RNA, which serves as a primer. A DNA clamp protein surrounds the DNA and attaches to DNA polymerase III, which builds the rest of the new fragment of DNA until it reaches the previously synthesized Okazaki fragment. When the fragment is finished, the polymerase releases from the Okazaki fragment. How are Okazaki fragments of the lagging strand joined together? A different DNA polymerase I, removes RNA and replaces it with DNA. However, it cannot finish connecting the Okazaki fragments. An enzyme called DNA ligase joins the Okazaki fragments together. Growth of the leading and lagging strands continues at both replication forks until there are two identical, double-stranded DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
So you might think that sounds sophisticated, but one thing one learns when one studies university-level biology is that just zooming in on one single of these proteins shows you just how brilliant a system this is. I'm just going to show one more animation about, this is a DNA polymerase. This is just one of the proteins that's responsible for copying the DNA molecule to synthesize a new strand. You want to just play this quick?
DNA polymerase faithfully replicates DNA by using the nucleotide sequence of the template strand, colored gray, to select each new nucleotide to be added to the 3' end of a growing strand, colored yellow. The different domains of DNA polymerase are colored differently. Before a nucleotide can be incorporated into DNA at the 3' end of the growing strand, the blue finger domain of the polymerase moves inward to correctly position the nucleoside triphosphate. A pyrophosphate group is released when each nucleotide is added. In this view, the details of nucleotide selection at the active site are shown, with the incoming nucleoside triphosphate and the template nucleotide in light blue. The growing strand is green and the template strand is red. When the finger domain moves inward, the nucleoside triphosphate is tested for its ability to form a proper base pair with the template nucleotide. When a base pair forms, the active site residues catalyze the covalent addition of the new nucleotide onto the 3' hydroxyl group on the growing strand, and the entire process repeats, at speeds up to 500 nucleotides per second. On rare occasions, approximately once every 10,000 nucleotide additions, the polymerase makes an error and incorporates a nucleotide that does not form a proper base pair onto the end of the growing strand. When this occurs, the polymerase changes conformation and transfers the end of the growing strand to a second active site on the polymerase, where the erroneous added nucleotide is removed. The polymerase then flips back to its original conformation, allowing polymerization to continue. As a result, such a self-correcting DNA polymerase will make a mistake only about once every 10 to the 7th to 10 to the 8th nucleotide pairs.
And indeed, one could go through many of these examples of these protein machines. They're all incredible just on their own, never mind when they work together in a cooperative fashion. So finally, let's just bash through some of the common objections to design, and then I'll wrap up and conclude. One objection, of course, is the God of the Gaps objection, which is to say that intelligent design is rendered simply a placeholder for that which we are ignorant of. But intelligent design, as I've already said, doesn't really work like that at all. It doesn't work that way. Intelligent design is not an argument from what we don't know, but what we do know about biology, and it makes a positive inferential case for design as the best and most causally adequate explanation of living systems. So I would say it's not a God of the Gaps objection. Another objection is that intelligent design isn't science. A number of the arguments for that: intelligent design violates methodological naturalism. Problem with that is that intelligent design doesn't necessarily actually contradict methodological naturalism, because intelligent design is a natural cause. I think it violates methodological materialism, but that's a different issue. Intelligent design is not falsifiable is another objection, whereas firstly, Karl Popper's demarcation criterion of falsifiability has actually been almost universally abandoned by philosophers of science. It's fraught with problems and difficulties. Theories in science typically aren't falsified by one piece of anomalous data. It takes a lot of evidence to slowly undermine and overhaul a scientific paradigm, for example. And then secondly, intelligent design actually is falsifiable because it's based on two premises. One, we find specified complexity in the cell. Two, specified complexity is best explained by design. So you falsify either of those premises and you destroy the design inference. Intelligent design does not make testable predictions. I gave you an example about predictions about protein folds, for example. Intelligent design does predict that you'll find specified complexity in the cell, so that's a prediction made by intelligent design. Intelligent design has no heuristic value, it's a science stopper. I think that's incorrect as well. Intelligent design, for example, can motivate us to look for example, function for junk DNA. And in fact, that prediction has been quite a fruitful one, and we've found as we look at the so-called dark regions of the DNA, the non-coding regions that we don't understand as well as other regions of DNA, that we've found that the general trend is that we find functions for the so-called junk DNA. Intelligent design invokes unobservable entities. But other disciplines, like particle physics, invoke unobservable entities too. Intelligent design doesn't address the modus operandi of design, but you don't need to have a mechanism or a modus operandi before you can infer design as the best explanation. In fact, we don't understand how mind interacts with matter, but we nonetheless know that mind does interact with matter and that it's a causal explanation in the world. Intelligent design, finally, isn't science because a judge in Dover said so, and of course that's referring to the famous Dover ruling in 2005 in Dover, Pennsylvania, where a judge ruled that intelligent design is unconstitutional because it's religion, not science. But since when was a district judge competent and qualified to make pronouncements on the philosophy of science? And the suboptimal design argument, you may have heard of the recurrent laryngeal nerve argument or the inverse wiring of the vertebrate retina. This objection is fundamentally weak because we infer design all the time from things which are suboptimally designed. And many of these, it's also a Darwinism that gaps argument because the argument has a retreat with each new advance in scientific discovery. And in fact, some plausible explanations have been found and elucidated as to why the retina is wired the way it is. And I think there's good reasons for that. And so I don't think it's particularly a persuasive objection to intelligent design. Then there's the incremental indispensability objection to irreducible complexity, which is basically it says that we are left with an illusion of irreducible complexity because how can you rule out the possibility that modification or even loss of components can render other components that were previously only helpful essential? Behe put forward the mousetrap as basically an illustration for irreducible complexity. You have a base, you have a hammer, you have the spring, the holding bar, and so on. And each of these parts is necessary in order for the mousetrap to perform its function of catching mice. One way of responding to the argument of incremental indispensability is to suppose that you evolve from a mousetrap into a rat trap. And so you duplicate the spring, let's say, you have two springs. And imagine that the first spring became lost or modified or in some way, and so it became non-functional. And so the second spring, now it was previously only helpful, now it becomes essential. But you nonetheless still need something performing the function of the spring. The specific identity of the spring or the thing performing the function of the spring isn't necessarily what's important here. And so I prefer to define irreducible complexity not in terms of structural components, but rather in terms of functions. You need a certain number of functions in order for the system to perform its job. Finally, Ken Miller, Brown University biologist, put forward an objection to the flagellum argument for irreducible complexity. Basically, he argued, well, there's the type 3 export apparatus in the flagellum that's involved in flagellar assembly, and it's also found outside the flagellum. It's called the type 3 secretion system. It injects toxins, poisons into multicellular organisms. Famously, Yersinia pestis, the causal agent of bubonic plague, has one of these systems. Salmonella uses these to inject toxins and so on. And there's a number of problems with this argument. He argues that the flagellum evolved from something like this. But did the flagellum evolve from a type 3 secretion system? Well, first thing to note is that the type 3 secretion system actually postdates the flagellum. The flagellum predates the type 3 secretion system. And there's a number of different lines of evidence for that. It makes sense evolutionarily as well, because you need to be able, there's an evolutionary pressure to be able to swim before there's an evolutionary pressure to be able to inject toxins into multicellular organisms. And there's some good molecular evidence, I think, for this notion as well, this thesis. Also, there's some reason to think that the type 3 secretion system is itself irreducibly complex. In fact, a 2001 study, quote, examined the effect of loss of function mutations in each of the type 3 secretion associated genes encoded within SBI1, that's in Salmonella, on the assembly of the needle complex, finding that many of the type 3 secretion system components are required for the system's function. So finally, let me just bring this to a conclusion. Intelligent design is based on standard principles of scientific reasoning about the past, as I mentioned, the historical abductive method of scientific reasoning. Intelligent design challenges the concept of unguided chemical and biological evolution. The design inference is based on cutting-edge advances in molecular biology, which reveals the complexity, elegance, and engineering prowess of cellular machines. And thank you. I'll be very happy to take questions.